It's a pleasure to introduce Paula Hammond. Paula Hammond is the executive officer of the department, which means that she has the unenviable task of convincing people what to teach and negotiating space, which shows you something about her diplomatic skills. She's also the bio professor of chemical engineering, and she was recently a fellow of the American Institute of Medical and Biological Engineers. And also recently she was elected as Distinguished Scientist of Harvard University. And with that, Paula?
Thank you. So I'm just extremely excited to be able to talk and celebrate Ed today. I have to say that after listening to the talks this morning, I was especially excited about the fact that I was hearing things that I myself had experienced from people who experienced it 20 and sometimes 30 years before me with regard to the impact of Ed Merrill. I wanted to share some of the things that Ed Merrill shared with me. As you can see on the left, I too was highly impacted by the Cheshire Cat analogy. That was one of the things that really fascinated me about polymer science. And the way that Ed would describe the Cheshire Cat and this vanishing effect, the ability to be able to probe on this level, just had all of us on edge. If you were to look at the classroom while he was lecturing, it was as if you were at a show and people were really listening for what happens next. He had us on edge. He really had us enthralled with polymer science. But I have to say, this was in graduate school, and I experienced graduate school in 1989, 1990. But my very first encounter with Ed Merrill was as an undergraduate here at MIT. I was here from 80 to 84 working on my bachelor's. I was doing my chemical engineering work. I thought, this is great. But when I entered, I thought I'd be playing with molecules. And here I am. I'm doing a lot of thermal and a lot of kinetics and a lot of continuum sorts of approaches. Where's the molecule? And just as I was beginning to get a little distraught about seeing my chemistry again, I took Ed Merrill's course as an elective as a senior and got really excited about polymer science and decided in the back of my mind, this is what I'm going to do. So I eventually went back for graduate school to get my PhD at MIT. I learned from Ed Merrill from the Great Book of Flory, all of my polymer physical science. And I also learned polymer synthesis from Rimp and Merrill. And when Ed taught this class, I was especially excited. And I do use polymer chemistry as one of my major tools. But another thing that Ed shared with me was actually the opportunity to motivate and to teach on these subjects and to excite others about the field. And I actually co-taught TIM 467 with him. It was TIM 67 and transformed into TIM 467 as we brought sort of the undergraduate level into the class. Ed Merrill did this by introducing a range of very approachable experiments that could be done with very simple equipment. And during that time, we also transformed Building 31, the combustion lab, into the Polymer Science Teaching Lab. This is a picture of it while it's empty, without all of the things cluttering the bench tops. Looks quite different now. I also began teaching TIM 569. It was TIM 69 when I took it. We introduced the 5 to indicate it as a graduate level course, and this is the polymer synthesis class. So I've had the honor of working with Ed Merrill and teaching with him and learning how he excites and motivates students. But I was also inspired by the work that Ed Merrill was doing and his research interests, which ranged from things at the surface, including tethered polyethylene glycol chain molecules. These are some of the things I actually knew Susan Sophia. We were in our lab right next door to each other when I was starting up my research that really fascinated me about controlling protein adsorption. I was also fascinated by the synthesis of these star molecules and these other very highly controlled architectures that Ed Merrill began to work with and to probe. And I'm going to talk about how those interests have motivated me as we move along. But I'll also try and give an example of how even his work on orthopedic implants has actually, in a strange way, come back around. So in our lab now, we're working on a problem related to this. And I should mention that we are in the same lab that Ed Merrill occupied in Building 66. We're in 66525 and 553. And we were able to benefit from Ed's presence in that lab. Even now, when we do our major lab cleanups, we'll always find a small vial of PEG 2500 or something like this, written, 1990 or 1980, and we can still uncover those. It's amazing. We have a lot of PEG in our lab, and we're preserving some of it for posterity. So I'm going to try and describe two different approaches that we use in our group to examine biomaterials. And in this first example, we're really interested in being able to address multivalency in the targeting of nanoparticles. In this case, the objective is to use a nanoparticle to carry a chemotherapeutic drug through the bloodstream into the tumor. A number of these things can be done by using nanoscale particles that encapsulate the drug because enhanced permeation and retention effects can help to entrap that nanoparticle inside the tumor. The tumor has leaky blood vessels. Those leaks are about the size of nanometer scale holes. And in our case, if we can design materials that are within that size range, they will get captured in the tumor for some period of time, 18 to 24 hours, for example. However, for us to actually get these into the tumor is a different story. If we've learned our lesson correctly from Ed Merrill, when we're introducing these into the bloodstream, we've decorated the nanoparticles with polyethylene oxide, something that can help prevent the particles from being taken up by the body's immune system or macrophages. And in doing so, we actually allow that system to act as a stealth nanoparticle, which doesn't get recognized by the body as a foreign material system. However, if you've done that well, when we get into the tumor, its interstitial regions, there's not a particularly good reason why a tumor cell would begin to take up these nanoparticles. Molecular targeting has been used to specifically target the uptake of nanoparticles. In this case, we use a very specific ligand that interacts with a receptor that is overexpressed by the tumor cell. Now, this is a very interesting and very exciting area. It's grown a great deal. But a number of people have really examined the number of ligands and the impact of the number of ligands. We're interested in a slightly different question, which has some biological impact, which is whether or not the way we arrange those ligands on the surface can also have an impact on the degree of uptake that these particles have with cells. So is the arrangement of ligands as important as the density of ligands on the nanoparticle surface? For us to be able to probe this, we couldn't use our usual trick of a linear block copolymer because we actually allow this ligand to explore a large amount of space. So these are roaming around if we have a sufficient length of PEG, and therefore very difficult to contain in specific clusters. We also have an architecture which doesn't allow us to confine them effectively. So what we're actually doing is using a block copolymer architecture in which one of the blocks is linear and the other one is systematically branched dendron, so that we can actually use the dendron to present the ligand in very specific predefined clusters. We then mix that with a block copolymer which does not contain ligand, and we can gain control of the spacing of these clusters and their number density on the surface. So this work, which was primarily done by my student Ji-Yeon Poon, involves this concept of molecular design. And again, one of the things that excited me so much about Ed Merrill's lecture was the fact that you can control what your macromolecule is going to do with very large amounts of precision. Here we're using N-carboxy anhydride chemistry to generate a linear block, which is polybenzoyl aspartate. This is one that we chose specifically because linear block copolymers with this block are already in clinical trials 3. We wanted to see if we could start with something that was sufficiently of interest to the clinical community. We actually initiate this with a dendron. This is a polyester dendron. It has this very systematic branching architecture. And at the end groups, we have 16 of these end groups. We have polyethylene glycol, fairly low molecular weight, 600 grams per mole. and we can attach ligands via this carboxylic acid group at the end. The ligand that we're studying here is folic acid, or folate. Folate actually is something that can be used to target a broad range of gynecological cancers. The folic acid receptor, or folate receptor, is overexpressed by ovarian cancer cells, among others. So we're using as our model KB cells, which are ovarian tumor cells. So I'm just giving you a brief summary of what we found in this approach. We actually designed these systems so that we had cluster sizes ranging from 1 on this G16 dendron out to the full 16 N groups, and refer to that using 0 to 100% functionalization. However, we, in this particular set of experiments, varied these so that they were mixed with non-functionalized systems, so that the net number of ligands on the nanoparticle surface was essentially the same, about 2,000 to 2,500 folate groups on average on a nanoparticle surface. So the number of ligands is the same, but the way in which they're presented is different. And as we move up, with the exception of the 0% folate, we're looking at the mean cell-associated fluorescence. We see that there's very little uptake after 24 hours in KB cells with these non-functionalized systems. However, when we begin to functionalize, we see this uptake increase. What we observe is that there's a specific optimum which occurs at 20% folate. That's about 3.2 on average folate per dendron. And then we begin to see a systematic decrease in the amount of uptake as this cluster gets larger and larger. So what we're finding is that sheer number doesn't really impact beyond a certain point. We've done several experiments where we've looked at this with varying amounts of ligand density. However, the way in which ligand is presented can have an extreme effect. So this is, again, we've done several experiments in vitro. And if we sort of think about what's happening, we believe that we have a dendron which actually presents an area to the cell membrane surface, which is roughly 50 to 80 nanometers squared. These are micellar systems. Because they form these micelles, we can think of this as a roughly average 90 nanometer micelle. There's going to be some curvature effects, but if we extrapolate, we have an area that is big enough to take up about 5 to 6 of these folate receptors based on their estimated size. When we just have singular functional groups on these dendrons, we're really not optimizing the amount of interaction that we can get, the number of binding, active binding events that we can get in these systems. As we begin to increase, we see that the systems in this range between the 20 and the 40% folate were the highest bars. They fall in this range of 3 to 6, give us more optimal binding. And when we get beyond that, we begin to see steric effects, binding hindrance, and binding exclusion effects due to the larger cluster sizes. We also think that there's a tendency on the cell membrane for these folate receptors to pre-cluster. They float on lipid rafts. There's been some imaging of the cell membrane surface. And it's been reported that these tend to cluster in groupings of about 3 to 4. If that's true, that may explain why our 20% or our 3-cluster group actually gives us the best performance out of all of these, as opposed to the higher cluster size that fits within this range. So we can actually look at this in vivo. We've done fluorescence molecular tomography to do this. We have on the right flank of the mouse a tumor that's a KB tumor, so it overexpresses the folate receptor. On the left flank of the mouse, we have a non-folate presenting tumor. And as we can see, as we move from 0%, this is after 24 hours, so we're beyond the enhanced permeation and retention effect at this point, we see very little accumulation. With the 20% folate system, we actually see, in these yellow and blue colors, indications of much higher amounts of concentration of the drug nanoparticle in the tumor. If we actually make these clusters a little larger, here we're going to the 60% range, we see that we've gotten a decrease in the amount of uptake. And finally, we get to cluster sizes that are too large to allow sufficient binding for uptake. We also have done a great deal of quantitative analysis, in which we can get direct comparisons. And again, we see the same effect, that the 20% system is the one that gives us the highest uptake. We've actually begun to look at these systems with respect to tumor regression when we encapsulate a hydrophobic drug like paclitaxel. In this case, what we see is that compared to saline, and here we have the no treatment group and the saline group, we end up with a very high rate of tumor growth. We compare that to our targeted vehicle, which has 2.5 milligrams per kilogram, and we see a much lower response. Our system, when we just introduce the untargeted treatment, does give us some degree of remediation due to that enhanced permeation and retention effect, but it's much more pronounced when we use our molecularly targeted system. And here you can see a comparison between our targeted system and free paclitaxel administered to the mouse at the same dosage. We've also done cytotoxicity studies with these systems, and we found about 2 orders of magnitude lower cytotoxicity in our targeted system compared to the free paclitaxel. So we're continuing to examine this. We're very interested in using these concepts of molecular design to further these systems. We're now beginning to look at much more specific peptides that are going to target either the vascular wall of the tumor or specific tumor cells. And this is part of an ongoing effort within the Koch Institute for Integrative Cancer Research. So I've talked about drug delivery and the very specific synthetic design of a macromolecule. But we're also interested in using very simple self-assembly methods to generate drug delivery vehicles. And we're very interested in being able to deliver drugs from surfaces. So in this case, we use a technique called electrostatic layer-by-layer assembly. And this is based on simple electrostatic interactions. And I can truly say this is inspired by work from Alan Michaels, as well as the kinds of systems that Ed Merrill would describe in his, both in class and in his work in terms of polyelectrolyte effect in general. In this case, we do simply take an initially charged substrate and immerse it into a dilute aqueous solution which contains the oppositely charged polymer. We can then allow adsorption to take place along that substrate until ultimately the surface charge is reversed. At that point, electrostatic repulsion prevents any further deposition on the substrate. And we can rinse the substrate to remove anything that's not truly adsorbed. That monolayer that's adsorbed has a thickness that is controlled by the pH and the ionic strain. We're actually tuning the conformation of the polymer chain on the substrate from a very flat, tightly stitched, two-dimensional random coil to very dense and thick, loopy, shielded polyelectrolyte chain, depending on whether we've added salt or tuned the pH to decrease the degree of ionization in weak polyacids or polybases. Now we can adsorb a second polyelectrolyte on the substrate. It follows the same rules, and we can keep going back and forth, dipping and adsorbing. These layers tend to be anywhere from as thin as a few nanometers to as thick as tens of nanometers. And we can actually observe interpenetration of polymer chains down to about 1 to 4 layer pairs in these systems. And that was actually determined using neutron reflectivity studies, a kind of analogy of the Cheshire Cat in a way, the experiment that Ed Merrill was describing so long ago. You can also use hydrogen bond functionalities to have complementary polymer chains built on these surfaces. Now, why are we interested in doing this? It turns out that unlike vacuum-controlled, high-temperature, high level of equipment cost approaches to designing very thin films, we can create extremely controlled thin films, conformal surfaces at very low cost. And this allows us to adapt this very practical approach with the range of material systems that are charged or that can be charged. So I mentioned this dipping back and forth process. My students don't like very much to have to sit and hand dip these things. We do use robotic dippers. However, it's much more practical if we can speed this process up because we allow 10 to 15 minutes of absorption to get the surface to reach a kind of equilibrium state. So we've actually adapted this to an automated spray layer by layer approach in which we can actually spray using convection and misted droplets from a nozzle, each of these polyelectrolyte solutions in alternation with a rinse misting solution. So we're still introducing plus charge, a rinse, minus charge, a rinse. The alternation still exists, but now we're getting adsorption not from this very dilute system over a long time, but from this solution over a very thin film that is applied to this surface. So we end up greatly shortening the time for diffusion and adsorption. We've adapted this. My student has gone on to start a company based on this spray layer by layer technique because it can be used for a very broad range of applications. In our own work, we've used this to coat both flat and porous substrates. And this gives you an idea of an electrospun fabric from Greg Rutledge's lab, which we coated with a nanoscale thin coating. We can actually get each of these fibers individually coated, which gives us a great deal of flexibility. And we can ultimately get these films to bridge across and create a uniform film. So I mentioned an orthopedic application. We actually are looking at how we can actually introduce a range of therapeutics from the surface of an orthopedic implant. And we're especially interested in revision arthroplasty, in which you actually need to remove an existing implant. In the current procedure in the US now, they take that implant, it's suspected that there is some infection present, and for that reason, whether there is infection or not, the patient is braced up. They actually introduce antibacterial beads, sew the patient back up, and the patient remains braced for a period of 6 weeks or more, just to eliminate any potential infection. Then there's a second operation, the beads are scraped out, and a new implant is put into place. So this is the current US orthopedic approach. Quite expensive. We're very interested in being able to incorporate a very thin film that doesn't change the geometry of our orthopedic implant while delivering the amount of antimicrobial needed to eliminate infection. We're also interested in introducing growth factors which can allow the healthy regrowth of bone tissue following the release of the antimicrobial. So we are using layer by layer approaches because unlike conventional coatings, which may have many of these components mixed together, and for which you have a little less control of how they come out in the film, generally as a mixture, we have the promise of introducing these systems sequentially and letting them come out sequentially. There's another advantage to using layer by layer, however, and that's the fact that one of our components in many of these systems actually is the drug, meaning it's one of the charged species that we incorporate into the film. And in that case, we actually end up with very high drug loadings of these materials. So we've incorporated a broad range of different kinds of materials. We've looked at gentamicin, vancomycin, and a range of other antibiotics. I'm just going to describe what we're doing with the biologics very briefly. We've actually been interested in this in part because proteins are very sensitive to processing conditions. So being able to dissolve them in a traditional polymer matrix usually involves solvent or heat or both. And those things are things that can greatly reduce the activity. However, we're introducing very dilute protein solutions which allow us to maintain the activity of the protein during assembly. In the work related to the orthopedic implant, we're working with bone morphogenetic protein 2, or BMP2, and we're using an osteoconductive scaffold, which is just this beta tricalcium phosphate 3D printed. It's from Theryx. It gives us the ability to coat a very high area, and we can use our spray layer by layer technique to do so. We get a release which is fairly rapid over the first 3 to 4 days, and then a sustained release after that. We get very nice conformal coating of the implant. This is actually a model protein that's been fluorescently tagged so that we can observe it. This is just one of the images in a confocal microscope, but we coat inside the pores of these systems. when we implant these in vivo, we use this steric scaffold introduced intramuscularly into rabbits, and we can actually see these systems at 1 month and 2 month time points. This is a cross section, and this is a 3D reconstruction. We see bone growing. I'm sorry, these are mice. We see bone growing in these systems. We see it densifying, and we see signs of very mature bone. Here, the blue regions are bone, and this is our control, and we see that bone is growing throughout the scaffold. We also see over time the development of spaces which will ultimately become marrow. So we see signs that this is bone that is continuing to mature. Okay, so I have about 2 minutes left, I think. Maybe. Okay, I'll give one last example, which is related to this approach of using layer by layer assembly to incorporate a protein. And this is an approach which was done with my colleague Daryl Irvine. I should mention, as I was a product of the Program for Polymer Science and Technology, so was Daryl. He came back to MIT a little bit later as a professor in the Department of Biological Engineering and Material Science and Engineering, and he's interested in the immune response and the use of biomaterials for immunological applications. We actually were interested in layering films onto a flexible substrate that contained an antigen of some kind, and then applying it so that we could actually use it as a vaccine. And if I go back one, the idea is that then we would have the dendritic cells in the skin, the Langerhans cells, take up the protein and begin signaling T cells and B cells, the other immunological cells in the lymph node, to build an immune response. So in our early work, we coated a polydimethylsiloxane sheet with these layer-by-layer films. We would tape strip a mouse's ear so that we remove the stratum corneum, which is the most difficult layer to penetrate. And we place our layer-by-layer film on top of the mouse and then observe what happened. And in this work, we found that we're labeling using confocal ova red, and the Langerhans cells are labeled green. And as we go down from the top of the skin into the depth of the skin, ova penetrates very nicely into the mouse ear tissue. We also see that the Langerhans cells dwell at about this 10 to 15 micron and lower level. And we see signs that not only do these coexist, the protein and the Langerhans cells, but that the Langerhans cells at slightly lower depths actually take up the protein, and they appear to be carrying them. We've done experiments which indicate that these do move up to the lymph node. We've used a range of different approaches to determine that dendritic cells are activating B cells and T cells. Now we can go back to the spray layer by layer technique and use it to coat a fairly complex surface. We'd like to get beyond the stratum corneum, and one way of doing that is to use microneedles, a technique that was developed by Mark Prosnitz, who's now at Georgia Tech. So the idea here is that we can actually put down these thin films, insert them with the microneedle into the mouse ear or into the skin, and that when you remove the stamp or the microneedle, you will retain a thin film that will deliver the vaccine over time. Here we're actually using a very simple system, our degradable polyester, which is a positively charged polymer, with a negatively charged PLGA particle. It's negatively charged using a lipid layer. And this is an example of what these films look like when they're built up. You can see the particles quite clearly in these layer-by-layer films. So we've been able to actually coat these very effectively. And as you can see, you get coverage across the entire microneedle surface, as shown here. When you transfer them to the mouse skin, you actually deliver the entire film, and you can remove the stamp. And these films were released based on the choice of poly-beta-amino ester that we use, which determines the rate of release and the rate of degradation of these PLG nanoparticles. In this case, the nanoparticles contain DNA, and we use transfection of the luciferase gene to determine the efficacy of that transfection. At the top, you see our 24 bilayer film at 5 minutes. Then we have a 24-hour series in which we look at 1 bilayer, 5 bilayers, and 24 bilayers. So we're seeing dose dependence, and we see very strong transfection of these systems. In fact, they give us better transfection than a range of other very simple transfection agents. So what we've learned from this is that water-based assembly enables us to incorporate very sensitive biomaterials with unique function. We can actually combine different material systems using this electrostatic assembly approach. And it allows us, because it's a water-based system, to bring in biopolymers and biomaterial systems that would normally be fairly sensitive. And there are a range of these things which have applications from energy all the way out to the biological applications that we talked about today. So I'd like to congratulate and thank Ed for everything he's done to inspire this work and the work of all of us here. I think that it really requires the intersection between chemistry, engineering, and biology to do the kinds of things that are being done now. I acknowledge the students who did this work, who are shown here, highlighted in bold, and I really appreciate the opportunity to present for Ed today. Thank you.
Alan?
One question. Paula, very nice. That two-phase release of BMP2, you showed a rapid release and then a slow release. Have you measured the activity of the BMP2 coming out at first versus later? Is it equally active, or do you lose some activity after time?
That's a very good question. Because BMP2 doesn't have a very simple activity assay, we haven't been able to assay the BMP2. We have done this work with very simple proteins like lysozyme. And in that case, we see that we retain 95 to 100% of the activity throughout the release period. We actually can take a control protein and compare it to our protein that's released. In the case of BMP2, we only have the biologic response to use as a metric. We can do ELISA, and ELISA shows us that we have the protein is there, that we have all of the protein present, but it doesn't tell us if there's a very small change in some of those protein molecules. One of the things we'd like to do is a much more detailed study of this. Right now, the quantities that we're releasing, although they're very good for therapeutic applications, is very small.
Yep, right.
Very good question.